Okay, guys. I'm making these because, as I said in my email, a lot of you guys seemed to be confused about the DNA to proteins lab, which is totally understandable. It was a lot of text, it was a lot of reading, and if you didn't understand it, it made the whole thing kind of difficult. So I'm going to go through and I'm going to try to explain it to you verbally instead, so that you guys can be better prepared for your test, which I believe is Wednesday. Don't quote me, but I think it's Wednesday. So what I'm going to do first is, if you want to pull out your lab books, if you happen to have it nearby, I'm going to do lab 20 first, and then we'll move on to 20 and 22, or excuse me, 21 and 22. So hopefully that'll help you guys understand what's going on, rather than just, going all through it. We'll do the first one, and then we'll go to the second one, and I'll try my best to explain everything so that you guys can understand it for your exam. So lab 20 starts on page 50. And this is very much the sort of beginning information about DNA. We didn't really get to a whole lot up to DNA in class because this is when we were supposed to do it. So, DNA. DNA looks like a ladder, basically. It's kind of fuzzy. Looks like a ladder. And if you unwind the ladder, this is what it looks like. We have what's called a phosphosugar backbone, or a phosphate sugar backbone, which is the vertical part right here. So if you want to think of this as a ladder, it's the side of our ladder. And inside, the rungs of the ladder are made up by what are called nitrogenous bases, or nucleotides. This is your A, T, and G, and C. So as you guys have, most of you figured out just fine, A always pairs with T, G always pairs with C, always. And between the two sides of our ladder, these guys are held together by hydrogen bonds. Hydrogen bonds. Hydrogen bonds hold the two sides of our DNA together, which is really advantageous for us because when we are going to be using our DNA either to make more DNA through cell replication, or if we're using the DNA to make a piece of RNA so that the RNA can go make a protein, our two sides of our ladder actually unwind so that these two parts don't match up anymore. So it kind of ends up looking like this down here. And so our bases, our nucleotides, are in the middle. And inside that, the piece of RNA or other DNA, depending on what we're doing, is built off of that pattern. Hopefully that makes sense. So if this side over here is, we're using this side to make a new piece of DNA, this pattern over here is going to have the requisite pattern on the other side. So when we make copies of our own DNA, we can use that template to ensure that every piece of DNA is identical. Hopefully that makes sense to you guys. Now if you flip to the back of page 51, ask you some questions. We just talked about, DNA and A pairs with T and G pairs with C. All that's fine. I don't care that you guys, I don't know what Dr. Merchant does. For me, ATGC, you don't have to type out thymine or guanine or anything like that. As long as you know what letters go with which, I don't care. It's fine. So, and I know this example was in the quiz, but we're going to go through it. We're going to go through this part of it anyway. Okay, so we're using this top part of DNA, this strand right here. Ooh, that's a funky star. This part of DNA, we're replicating this. We're using this part of DNA as a template to make this new strand of DNA. So this is a new piece of DNA, and remember A always pairs with T, G always pairs with C. So this is what it looks like. G always pairs with C, A always pairs with T. Now, we could, in the future, use this part again to make another one of these, or we could use this part to make another one of these, because they're opposites of one another if you want to think about it like that. They match up, always. So if I were to give you a strand of DNA, just one side of DNA like that, you guys should be able to write the bases that pair up with them. Now this is one everybody seemed to have problems with. I'm going to write it sideways. One second. We had problems with this. And that's okay. It's okay. So what this is really asking us is, what are the steps that are used to make proteins? What's the thing that we start with that undergoes transcription? What do we get out of transcription? What goes into translation and comes out of translation? Because this is on a whiteboard and this is a camera, I have to write all of these letters backwards. So yeah, that's why it's taken forever. So this is how it works. DNA gets transcribed into a piece of RNA. RNA gets translated into a protein. And in the next video, we are going to look at these two processes a lot closer so that you guys can understand what they are and why they do what they do. Okay? That's enough for this one. On to the next one.